Well, hello, everyone. Good afternoon. Thank you for joining us for the virtual 9th Annual Forensic Science Symposium Lunch and Learn poster session hosted by the Global Forensic and Justice Center. We are recording today's sessions and we'll let you know when it is available on GFJC's YouTube page. I'll post a link to that in the chat for you to be able to access. Please use the chat feature to ask any questions that you might have. If there's any questions that we don't get to due to time constraints, they'll be provided in written format shortly. You can make this presentation full screen if you need to make it larger by clicking on the 4 arrows icon in the top right corner, and then click the same one to exit that full screen mode. With those housekeeping tips out of the way, here's Maurell Dsouza's poster and their presentation.
Well, good afternoon to everyone who's present here today. Here's my poster presentation. So this research was actually my master's thesis, and I just recently graduated from Florida International University with my master's in forensic science. This research was a collaboration with Dubai Police, and our goal was to help set up the practice of forensic entomology in the United Arab Emirates. Now, an easy way to do this with forensic entomology is to look at the pre-appearance interval as a way to estimate the post-mortem interval. So back in Dubai, we performed some field experiments, and we tried to build a statistical model for the same. Knowing the pre-appearance interval, the time of death for an insect-free corpse can be estimated. This is basically the time between death and initial colonization by carrion insects. So if any of you have not watched my video presentation earlier, just a brief introduction to my research. And if you have any questions along the way, feel free to put them in the chat and I'll try my best to answer all of them. So just an overview of the experiment that we performed. We set up some rat carcasses in the field in Dubai, and over different time periods, we pulled the rats back into the field. And this was just a way to study the succession pattern of carrion flies that colonize an insect as a corpse is decomposing. One thing to mention here, which is important, is species identification. For such statistical models to work, we need to ensure that we're working with the right species. And so for this study, since we were working with sarcophagids, we used mitochondrial DNA sequence data to identify the species we were working with. And the methods we used were, DNA extraction, PCR amplification, purification, and finally we sequenced our data and used the POP software to make our specimen identification, where our unknown species were compared to a reference database. And if you look at the observation table here, this is the occurrence pattern that we built. We happened to find 2 different types of sarcophagids in Dubai. One had this spotted abdomen pattern, which you can see in this image, and one had a more checkered appearance. And we also looked at Chrysomya albiceps. This species is commonly used in the United States and European countries, but we found that they were too slow to colonize our rats. And maybe I'll explain that a little more in the next phase. But how this works is, this column here is the time interval. So day 0 is the day of death, and each row represents a different time that the rat was picked from the field. And this value here represents whether or not the species was present on the rat carcass. And we used 9 rats, and that's what 9 is. This is a 9-rat table. And this is how we build the statistical confidence limit for at the 90% level. And for identifying the pre-appearance interval, we need this value here to come down to 0. So either 0 or 1 for 90% confidence limits. And you can see Chrysomya albiceps never really came down to 0. And that's why we couldn't use it for our study. So we focused on the 2 sarcophagid specimens that we had. And so this shaded region here is the pre-appearance interval. So what that means is if you encounter an insect-free corpse, it's probable that that corpse was exposed within this time interval here. Or in other words, if you do find a corpse with that insect species, it was exposed for longer than that time interval. So that body was present at the crime scene for longer than this time. Another thing to mention is that For this to work, every single checkered abdomen sarcophagid had to be one species, and every spotted abdomen sarcophagid had to be one species. If you look at table 2, what we actually found out was that we had 2 checkered abdomen sarcophagid species and 2 spotted abdomen sarcophagid species. So it's double of what we were expecting. But we were able to confirm the presence of Wolfhartia nuba, this species, in every single rat carcass that was used in the study. So because of that, we can confirm that the spotted abdomen sarcophagid occurrence pattern, this pattern here, could be confirmed as that of Wolfhartia nuba. Another thing that's really good with this type of research is that it can be used at any sex of the fly and at any life stage. And so this can be used for further implementation of forensic entomology in the United Arab Emirates, and also for the other 3 species that we discovered. So that's just a brief overview of my entire research. And if you have any questions, I'd be happy to answer them.
Thank you, Maurell. That was really good. If there are any questions, please feel free to type them in the chat, and we can get those answered for you. We did have a few people enter while you were speaking, so if any of those folks have questions that maybe they had missed, feel free to let us know that in the chat, and we can go back over that.
Yeah, if there's something that was maybe rushed over or you didn't understand, let me know. I'll go over that one more time.
Also, all of Maurell's contact information, including the video that she mentioned, is all in the chat as well, that you can go and watch that video or contact her if anything comes up after this session.
So there was a question in the chat that asked, was there a difference in the time arrived for the different species within the same morphotype? So it's difficult to answer that question without looking at all of the data. There's a lot of, so this table that I have here on the slide is actually one out of many tables that we have. But I guess the simple answer would be yes, there was a difference. For example, okay, so how it worked was after the rats were collected from the field, they were held until adult flies emerged from them. And so while I did my specimen identification, I pulled out one spotted and one checkered fly from each of the rats, right? And if you look at the occurrence of the species, I picked out about 150 something Wolfhartia nuba and only about 50 Wolfhartia indigens. These are the 2 species I'm talking about in the spotted abdomen. So just by that estimation, I could personally say that there was a significant presence of Wolfhartia nuba in comparison to Wolfhartia indigens, which could mean that Wolfhartia nuba colonized faster or more frequently than the other species. They were almost the same, almost equal amounts of Ruficornis and ducks. So it's difficult to make that prediction with the checkered abdomen. But I could say that for the spotted abdomen, that we saw one species more predominant than the other. I hope that answers the question. I guess I could also add that if you look at this table again, you can see there's a difference in the time between different species as well. As I said, with Chrysomya albiceps, they were too slow to colonize these carcasses, which is why we couldn't use them for the study. But also, there could be an ecological reason why these species colonized slower than the other 2 species. A little bit about the ecology or the life of these insects. Chrysomya albiceps maggots, they actually feed on other maggots. So it's possible that they colonize slower than the sarcophagid species because the maggots eat the sarcophagid maggots. So it's probably why they're arriving later. So we're not very sure what's going on with the flies and why they colonize at different times, but it has been observed.
Great question. If anybody else has any questions, feel free to put those in the chat. And you can either send it to me as the host, or if you have the option to send it to Maurell, you're welcome to send it to her directly as well.
Yeah, I received another question actually privately. The question says, what environmental conditions were considered? So that's a really good question actually, and that's something that we are trying to look into as well. for those of you who are not very familiar with the temperature and environment in United Arab Emirates, it's extremely hot in the summer and it gets really cold in the winter. This study was performed in December, where it was much cooler. So we actually have record of the weather on the days that the experiments were carried out. Oh, we did repeat the experiments 3 times for 2 fields. We had 2 fields used for our study. So at both fields, we recorded the temperature. But there are talks going on about like redoing the study in the summer months, because it's much hotter than the winter months. So we don't have a definite answer to whether the pattern will change for the summer, but I believe it might. I have another question. So question says, regarding the generalization of these results to human remains, would we expect different species to visit humans versus rats? And would the colonization times be different between rats and humans? And she asked this because there is research going on that shows that blowflies colonize humans at different times than pigs and rabbits. So that's something I should have mentioned as well. So these results are definitely useful to the extent that it can be applied to humans. We have been using rats as models for humans. And so it's most likely that it will work for humans. Also, keep in mind that this table is at the 90% confidence limit. So we can say with 90% confidence that these studies will work. And that's why we have such a large sample size. We used 216 rats to build this table. This is only one out of the many tables. I'll repeat that again. It's just an example of the many tables we have. This is representing a 9-rat table out of the 216 rats that were used. Another challenge with this question is, how do we obtain 216 humans for this study? It's a little challenging there. So we had to, deal with rats. But yeah, that's something that would require a lot more research to confirm if our results are identical between the different models used for the study. Due to logistical, financial reasons, we couldn't use humans or pigs or rabbits for the study, and we had to settle for rats. Okay, we have another question. Was there any considerations given to the type or the composition of soil in the field, and what insects other than that was considered in the study could influence the study? Again, that's a really good question as well. So a little more detail about my research. When we performed these experiments in Dubai, we worked with 2 different fields. One was in hyper-urban location in the middle of the city. We didn't have soil here. The ground was sand, like Dubai sand, white sand. And our second site was an abandoned palm plantation where there were much more trees in the back. And again, it wasn't really soil, but it was more, I want to say rocky surface. So we worked with different fields in Dubai. And we have all of the data for the 2 different fields and how the succession pattern changed. And if you would like, please pass me your email and I can give you a more detailed explanation about the 2 different sites, if that's okay with you. So I can go in, like, it's difficult for me to pull up all the tables now and discuss that with you. But yes, we did work with 2 different soils or 2 different settings in Dubai. And other insects. So we ended our experiment after 3 days. As you can see in the table, we had day 0, which was the day of death, day 1 and day 2. So we ran our experiment for 3 days and we didn't take the study any further. And we looked at these 3 species, Chrysomya albiceps, the checkered abdomen sarcophagid, and the spotted abdomen sarcophagid. These were the ones that were predominant in our studies. We didn't see any other species coming in. Besides the, the beetles and ants that walk around, these were the carrion insects that we saw on our carcasses. And so we decided to stick with these 3 because they were more abundant than the others. I hope that answers your question too. I'm also going to add one more thing. So this table only works for 1 species. So if you're using this occurrence pattern for Wolfharsha nuba, for example, as I'd mentioned, that we confirmed the spotted abdomen sarcophagid occurrence pattern for Wolfharsha nuba, this cannot be overlapped with the other species. It is independent to 1 species alone. If you want to try to develop a study or build a study for more than one species, you would have to increase your sample size. So for one species, we use a 9-rat table. If you want to do 2 species, you'd have to do 11-rat table. And if you want to do a 3-species study, you'd have to do 26-rat table. So you can see that the sample size increases dramatically. And that's why we stuck to 1 species. I think I might just do another brief run through with the research just for the people who are just joining. Okay, for those of you still joining in, my contact information is in the chat. Please feel free to contact me with any questions, comments. I'd be happy to respond to all of you. So just putting that out there because I got a question regarding that. So again, thank you for tuning in to my poster presentation. I'm going to go over it one more time if you just joined the group. This is my master's research that I just graduated from FIU with my master's in forensic science, and this was the research that I did. This research was a collaboration with Dubai Police in the United Arab Emirates, and it was funded by CARFS, the Center for Advanced Research in Forensic Science. The main goal of my research was to help establish the practice of forensic entomology in the United Arab Emirates. And the most common way to use forensic entomology is for estimation of post-mortem interval, which can be done by looking at the pre-appearance interval pattern. The pre-appearance interval is simply the time that it takes for an insect to colonize a corpse, a decaying corpse, when it is exposed for colonization. And for such a study to work, we need to ensure that we're making accurate species identification. And since in this study we were using sarcophagids, I looked at mitochondrial DNA sequence data because it's difficult to identify sarcophagids by their morphology. It's possible that different species appear almost identical with sarcophagids. And so we looked at mitochondrial DNA. A little bit about the field work that we performed. We had 2 different fields in UAE. We had one that was a more hyper-urban field and one that was a more rural field. It was an abandoned palm plantation. And what we did was we laid out rat carcasses and we collected them after different time intervals. So this is part of a succession study to see the different species that colonize the carcass. And to make my species identification, I extracted DNA from the legs of adult fly specimen, and I then amplified them using the Promega master mix. The primers I used amplified a 600 base pair region of the cytochrome C oxidase 1 gene of the mitochondrial DNA. And this is the common gene that's used for species identification. After purification, using the QIAGEN kit, we outsourced our sequencing to Macrogen in Korea, and our final identifications was made by using the POP software. So all that did was compared our sequence data, the unknown sequence data, to a reference data set. That data set was built by it consisted of specimen or species from the Middle Eastern region. So all of the sarcophagids that we could see discovered and published from the Middle East, those were used as our references. So if you look over here at table 1 in the observation section, this is the occurrence pattern that we had. This is just an example of many other tables. And this is at one location in Dubai, which is PA, our abandoned palm plantation. And this column here represents the time interval, day 0 being the first day of death, and each interval after that when we collected the rats from the field. We observed 2 different morphology of sarcophagid, one with this spotted abdomen pattern and one with a more checkered abdomen pattern. And if you look at these values here, they represent whether the insect species was present or absent on the rat carcass. So this table represents 9 rats. So 9 represents that 9 rat carcasses were used to build this table. And it tells you whether or not Chrysomya albiceps, the species here, is absent or present. And to reject a time interval, we need that value to come down to either 0 or 1. So you can see here 0 or 1. That provides you a confidence limit of 90%. And here the shaded region is the pre-appearance interval for this specific specimen. You can see here that Chrysomya albiceps never really came down to 0 or 1 in our 3-day collection period that we looked at. And so we couldn't use this species for our study. Chrysomya albiceps is commonly used in the United States and European countries, but as you can see from this table, it wasn't useful in the United Arab Emirates. So we focused on the 2 sarcophagids that we obtained. So this shaded region here is the pre-appearance interval. And for example, if you're given an insect-free corpse, you can say that that corpse was exposed within this time interval. So this time interval here. Or for this specimen, it would be this time interval. Or in other words, you could say that if you do find a corpse with that insect species present, it was exposed at that crime scene for longer than this time interval. And one thing to mention, which is important, is that for this table to work, all of these checkered abdomen sarcophagids should be one species, and all of the spotted abdomen sarcophagids should be one species. Or in simpler terms, that species should have been present on every single carcass that we used for this study. As I said earlier, sarcophagids appear to be identical even though they are different species. So that's something that we needed to confirm. And if you look at table 2, you can see that we actually found a total of 4 species, 2 for each of the morphotypes that we observed. To our good luck, we were able to confirm the presence of Wolfhartia nuba in every single rat carcass. And so we can confirm that the spotted abdomen pattern, which is this one here, can be confirmed as that of Wolfhartia nuba. This method that we use is extremely beneficial because it can be used for further implementation of forensic entomology in the United Arab Emirates, and also to confirm the pattern for the other 3 species that we discovered. And furthermore, it can also be applied to any of the sexes or at any life stage. Another common method that they use for specimen, like species identification, is by looking at adult male genitalia. But that process is extremely time consuming. So mitochondrial DNA was the better option for us. So that's just a brief overview of my work. If you have any questions, please feel free to put them in the chat and I'll answer all of them. If there's something you didn't understand, or if I ran through something too quick, again, feel free to put them in the chat. I'll go over it one more time if needed. Okay, I see a question in the chat that's asking, in the UAE, the identification for sarcophagid flies is done exclusively with mitochondrial DNA. The answer is no. Well, yes and no. Okay. This is the first forensic entomology related study that's being done with, in the UAE. And the other, like I said, the other way to identify sarcophagid flies is by looking at male genitalia. So you would have to pull out adult male genitalia from the flies, from the adult flies, and make a comparison with the key. But that's an extremely time-consuming step to, pull out the adult. Like, these flies are really tiny, so you would have to do it under a microscope, pull out all of the genitalia and compare it to a published key, and then make your identification. I had almost 200 flies that I used for this study, and it would have been extremely time-consuming and challenging to pull out every single fly's genitalia to make that identification. So the more easier and less time-consuming way to do it would be to look at mitochondrial DNA, and that's why we chose that method for this study. So all I did was extract the DNA, amplify it by PCR. I amplified the cytochrome C oxidase 1 gene from the mitochondrial DNA, and we looked at its sequence, compared it to reference sequences from the Middle East, and that's how we made our identifications. So no, it's not the exclusive way to make identifications, but it is the more easier way to do it. I hope that answers your question. So I see a question that says, in what genes do you see differences in the sequence between the species? So the gene I use to make my species identification is the cytochrome C oxidase 1 gene of the mitochondrial DNA. That's the common gene that is used to make species identifications. So in this research as well, after I got my sequence data from all of the flies, I had about 200 flies that I looked at. We made a comparison using a reference data set And we had about 9 species that were used from the Middle East. So we built a reference data set of fly species found in the Middle East, and we used the POP software. So it built a phylogenetic tree and made the analysis. So hopefully that answers that question. Also, the primers I used to amplify the gene was a 600 base pair region. So it covered quite a good area for us to confirm that the identification was accurate. I see another question, which I'm not sure how to answer. It says, is it correct in assuming that we are assuming that the PDF that underlies the observation samples is Gaussian, or is some other distribution assumed? I'm not sure I understand the question. So let me go over that table one more time. It's an occurrence pattern, and all that this table tells us is whether or not a species is present. So as you can see, we looked at 3 different specimens. One was the shiny green blowfly, or called the Chrysomya albiceps, and we had 2 different sarcophagid species that we thought were 2 species. Turns out they were actually 4 species, as we see in the second table. But here's what they look like. One was a spotted abdomen sarcophagid and one was checkered. And so this table represents 9 rats out of the total 216 that were used for the study. And so it just tells you whether the species is present or absent. And here is the column representing time, the time interval. So day 0 is the day of death. And we collected these rats over different time spans. So at the first day, whether the rat had that species or not is what we document. So day 1, you can see almost all of the rats did not have any insect species except one, which had a spotted sarcophagid present. So that goes throughout the time span. And then the shaded region is your pre-appearance interval. So how you reject a time interval is you need that value to come down to 0 or 1. And what that means is every single rat had that specimen present. As you can see here, all 9 rats had that species. And it's either 0 or 1 for a 90% confidence limit. And so, for example, if you encounter an insect-free corpse, you can say that it was exposed within this time region. Or in other words, you could say that if you found a corpse with that insect species present, it has been exposed at the crime scene for longer than that time region. So I hope that makes sense of what the table is. So it's just an occurrence pattern depicting this was a succession study, and this table is the occurrence pattern that we observed. And this is just one table out of many. We had we looked at 2 different sites, and we also looked at 2 different times of death, if you would call it. So we have the data for all of that. If you want to know a little more about that, feel free to contact me, and I'll try to give you a more detailed explanation about the differences.
Thank you. And Maurell's contact info is in the chat if you guys are looking for it. I just want to let everyone know we have about 10 minutes left in today's session. So if you have any questions, please put them in the chat so that you can get them answered. If we don't have time to get to them, we will provide those answers in written format shortly as well. So any questions, feel free to type those in.
Maybe I'll just one more time go over the brief of my research for those who have just joined, since we only have 10 minutes left. This was part of my master's research. It was a collaboration with Dubai Police and was funded by CARPS, the Center for Advanced Research in Forensic Science. We were trying to establish the practice of forensic entomology in the United Arab Emirates. And to do this, we tried to build a statistical model for the estimation of pre-appearance interval as a method to estimate postmortem interval. So the pre-appearance interval helps one to estimate the time of death for an insect-free corpse, because it's the interval between death and initial colonization by a carrion insect, provided that the corpse was available to be colonized. And so we ran a few field studies in UAE. We had 2 different sites that we worked at, and we had a total of 216 rats that we used. And what we did was we laid out these rat carcasses in the field, and over different time intervals, we pulled a subset back into the lab, and we observed them for the presence of different insect species. So it's a basic succession study. And for these studies to work, it's extremely important that you're making accurate species identification. And in this case, we used mitochondrial DNA to make our identifications because our useful fly turned out to be sarcophagid species and not what we anticipated. We were planning to work with Chrysomya albiceps, but they were too slow to colonize our rat carcasses. And I'll explain what that means when we discuss the table. So all I did was I extracted DNA, I amplified the cytochrome C oxidase 1 gene of the mitochondrion to make the identification. And our sequence data was compared to reference sequences using the POP software. And if you look at this table, this is the occurrence pattern that we observed. This is one example out of many tables that we have. This represents a 9 rat table, as you can see the number 9 here. And this was at one location in Dubai. And this column represents the time interval. So day 0 being the day of death, the day that it was exposed in the field. And to reject a time interval, we need this value to come down to either 0 or 1 for 90% confidence limit. And here's what I meant earlier when I said Chrysomya albiceps was too slow. This value never came down to 0 or 1, but it did come to 0 or 1 in the sarcophagid species, and so we continued to use that for the study. What 0 means basically is that every single rat was colonized by that insect species. So the shaded region here is the pre-appearance interval for that specimen. And so given an insect-free corpse, you could say that the corpse was exposed in that time interval, in this region here. Or in other words, if you do find a corpse that has this insect specimen present, then it has been exposed at that crime scene for longer than this time interval. And now for this to be valid, we needed every single checkered abdomen sarcophagid to be one species and every single spotted abdomen sarcophagid to be one species. But as you can see from table 2, we actually had 4 species in total. We had 2 checkered species and 2 spotted species. So that would mean that this table does not hold true. Fortunately, we were able to confirm the presence of Wolfartia nuba, that's this species here, in every single rat carcass that was used for this study. And so the spotted abdomen sarcophagid pattern can be confirmed as that of Wolfartia nuba. Also, this technique is really simple and can be used for further implementation of forensic entomology in the UAE. And it's also useful as it can be used to any sex of fly and at any life stage. So again, that's just a brief overview of this research. If you have any questions, you can put it in the chat and I'll try to get to them. If not right now, I'll have them written out.
All right, Maurell, I've gone ahead and locked the meeting so nobody else can join us. Amber, I think you are the Amber on the poster, so it's fine that you are here. Thanks for joining us this whole time. I appreciate it. Good job.
Perfect. Thank you so much for assisting me here.
Yeah, absolutely. Enjoy the rest of the symposium.